to the keynote lecture at this evening, and it's my great pleasure to introduce Martina Schüssler-Lenz to you. Recently, Martina Schüssler-Lenz was elected on her second mandate as the chair of the Committee for Advanced Therapies of the European Medicines Agency, EMA. The Committee for Advanced Therapies is responsible for evaluating the quality, safety, and efficacy of marketing authorizations of cell and gene therapies, advanced therapy medicinal products, ATMPs in short. Dr. Schüssler-Lenz received her medical degree at the Mainz University Hospital. Following this, she was trained in hematology, medical oncology, and completed an internal medicine residency and fellowship at Berlin Moabit Hospital and at Mainz University Hospital. She worked as a research fellow at Memorial Sloan Kettering Cancer Center and at the Instituto Municipal de Investigación Médica in Barcelona. Sorry for my Spanish. So in Spain, before she joined pharmaceutical industry for clinical drug development projects in hemato-oncology. Since 2005, she is working as a medical doctor at the Paul-Ehrlich Institute in the section Advanced Therapies and Tissue Preparations, where she has reviewed advanced therapies in clinical trials and marketing authorizations, and has provided advice to ATMP development and life cycle management. She is committed to facilitate the development of advanced therapies and has a specific focus in providing guidance to academic developers of ATMPs. Dr. Schüssler-Lenz has been a member of the EMA Committee for Advanced Therapies for many years before she was elected as its chairperson in February 2017 and re-elected for the second term in January 2020. The title of her talk will be Marketing Authorizations of Gene and Cell Therapies in the EU. Please, Mrs. Schüssler-Lenz, we are looking forward to your talk and ask you to share your slides with us.
Yes, I have provided the slides to your colleagues, so if I may ask them to put them on. Yes, this is very kind. So thank you very much, and thank you very much, Professor Nürnberg, for the kind introduction, and thank you very much for the invitation. So I guess that now we change gears a little bit in looking at marketing authorizations of gene and cell-based therapies. And if you move to the next slide, the next one, I've listed some of the keywords which you may have heard in the past couple of months or even in the last 2 weeks. Centralized European Marketing Authorization, European Medicines Agency, Paul-Ehrlich Institute, conditional marketing authorization, mRNA-based COVID-19 vaccines, viral vector-based COVID-19 vaccines, US FDA. So I try in the next 30 to 40 minutes to bring a little bit clarity, hopefully, in our quite complex regulatory system. And I will talk about the authorization of innovative medicines in the European Union, about the centralized marketing authorization of gene and cell-based medicines, give you examples of gene therapies, and then conclude. On the next slide, you see the two main classes of medicinal products. Medicinal product, a substance or a combination of substances that is intended to treat prevent, diagnose a disease, or modify physiological function. And you see here the one is the chemical group of medicines, and the other is the biological group of medicines. And a representative for the chemicals is remdesivir. Chemicals are also called small molecules. And for the biologicals, we have here erythropoietin, Herceptin, a monoclonal antibody, and then even the more complex products, Yescarta and Chimeric CAR T cells, and we will have a look at these in a minute, and Luxturna, a recombinant adeno-associated viral vector. And these are lower parts, CAR T cells, and are called advanced therapy medicinal products. Allow me on the next slide for those that are from the German region to give a short overview of the responsibilities for chemicals versus biologicals in Germany. So for the chemical medicines, the responsibility is at the BfArM in Bonn, the Federal Institute for Medicines and Medical Devices, which also takes care of some biologicals like recombinant proteins. The Paul-Ehrlich Institute in Langen, close to Frankfurt, is responsible for monoclonal antibodies, advanced therapies, vaccines, and others. So in Germany, we have the special situation that we have two national competent authorities, or competent higher federal authorities, as they are called, below the Federal Ministry of Health, that are responsible for medicines. On the next slide, I have outlined the process of medicinal product development from research discovery through marketing authorization to post-authorization safety and efficacy monitoring. And we divide this life cycle of medicinal product into these three phases. And you see here the preclinical and the clinical development are part of the pre-authorization phase, which you can divide further into the discovery phase, the non-clinical evaluation, the manufacturing of the product for the clinical trial, and then you see phase 1, first in man clinical trial, phase 2 and 3 trials, which then result in a marketing authorization, hopefully, which is then followed by the post-marketing phase. So these are the three main phases and the life cycle of medicinal products. And on the next slide, you can see that this is quite a complex situation in Europe. For example, compared to the US, where you have the FDA, we have in Europe different responsibilities for different stages in the medicinal product development. And so you can see here again, about in the upper part, pre-authorization phase, authorization, post-authorization phase. Then below you see the assessment of quality, safety, efficacy, which is then results in a benefit risk assessment at marketing authorization, and is then followed in the post-authorization phase by the monitoring of the safety and efficacy of the medicine. The national assessors and experts from the European Union member states are involved in all these stages, but the pre-authorization phase is more in the responsibility, or mostly in the responsibility, of the national competent authority, while the authorization phase is in Europe in the responsibility of the scientific committees at the level of the European Medicines Agency. This means an applicant or a developer of a medicine, of a gene therapy, let's say, when he is in the pre-authorization phase, he submits the dossier to get the clinical trial approved to the national competent authority, like BfArM, like Paul-Ehrlich Institute, like ANSM, Agence Nationale Sécurité Médicaments, that would be the French part then. But when he has done his preclinical and clinical development and wants to submit a marketing authorization, this marketing authorization is then submitted to the European Medicines Agency. And on the next slide, yeah, you could ask now, why is this? I mean, for the COVID-19 vaccine, for one of the COVID-19 vaccines, the product has been developed in Germany, clinical trials have been performed in Germany. Why don't we approve the this type COVID-19 vaccine on a national basis? And this is because most products, most medicines, and the innovative ones have to undergo the centralized procedure at the level of the European Medicines Agency. And you see here that there is a legal background for this, in that medicinal products developed by means of biotechnological processes need to go through the centralized procedure. Advanced therapy medicinal products do, and medicinal products with new active substances for AIDS, cancer, neurodegenerative disorders, diabetes, autoimmune disease, viral diseases, and orphan drugs. So these have to go through the centralized procedure. And on the next slide, we have a closer look now at what my specific responsibility is, the advanced therapy medicinal products. We call gene and cell-based products in Europe advanced therapy medicinal products. And in the meantime, US FDA has also adapted this term for gene and cell-based therapies. And you can see here that we have this regulation now since 11 years. ATMPs are medicinal products, are authorized in the European Union via the centralized procedure. The principles of the existing legislation apply as for other products, quality, safety, and efficacy assessment. But a specific committee, a specialized committee, has been put in place in 2009 when this regulation came into force, with experts and specific expertise in gene and cell therapy. And that's the Committee for Advanced Therapies where I work. And you see below the three groups of ATMPs. On the right side, the tissue engineered products. In the middle, somatic cell therapies, and then on the left side, gene therapies. And gene therapies are these products that contain a recombinant nucleic acid, which leads to the therapeutic effect in the treatment of the disease. And yeah, now you can ask, well, then what are the mRNA and vector-based vaccines to protect against COVID-19 also gene therapies? And the answer is no. In principle, they are, but the decision was taken on the legal, in the legal framework that gene therapy medicinal products shall not include vaccines against infectious diseases. So these are excluded from the gene therapy definition. On the next slide, you see the overview of the marketing authorization procedure. So for a product that is not an ATMP, not a gene and cell-based therapy, that is, for example, a COVID-19 vaccine, that this medicine is evaluated by the CHMP, the Committee for Medicinal Products for Human Use. If a product is classified as an ATMP, it is evaluated by the Committee for Advanced Therapies, which you see on the left side. That means the quality, safety, and efficacy assessment resulting in the benefit risk assessment is conducted at the level of the cut, usually by two teams from two national member states that we call rapporteur and co-rapporteur, that bring their assessment to the committee, where we meet once a month, we discuss and then decide. And on the next slide, you see a little bit more details about the marketing authorization process for all centrally authorized products, which starts with the dossier submission. So the applicant, the developer, submits the dossier. The dossier it contains all the information: quality, safety, efficacy, and post-authorization plans. The European Medicines Agency colleagues in Amsterdam validate the dossier. And let me say here, the colleagues in Amsterdam at the European Medicines Agency, these 700 or 800 people, their task is to coordinate the scientific assessment. They do not do the scientific assessment. As I've said, the scientific assessment is done in the teams on in the national agencies, but the task of the EMA, and that's a very complicated and complex task, is to coordinate the process. They also validate a submitted dossier, and then on the evaluation is done in the scientific committees, CUT or CHMP, CUT and CHMP, and start with day 1 and goes until day 120. So the first primary evaluation phase is 120 days. That means the 2 assessment teams have 120 days to assess quality, safety, and efficacy. Then it's discussed. Questions are posed to the applicant in a clock stop. On day 121, the clock starts again, and then we have time to review additional data until day 210. So it's a total 210-day procedure with additional clock stops, and usually extends over a year or more than a year, this assessment procedure of a marketing authorization. The result is a benefit risk assessment, and in case of a positive benefit risk assessment, a positive opinion at day 110 by the CUT, confirmed by the CHMP. And then, and that I showed that on the previous slide, the evaluation report, and sorry, it was on the previous, if you go one back, yeah, on the previous slide, you see here, this opinion is then passed on to the European Commission to do the authorization for placing the product on the market in the European Union. That's the task given by the European Commission based on the assessment report of the scientific committee. If you go to the next slide, yeah, you see that this then, the decision of the European Commission results in 1 marketing authorization, assessed, supported, valid in the entire European Union, including Norway and Iceland. On the next slide, you see a little bit more in detail the tasks and composition of the CUT. Regarding the tasks of the Committee for Advanced Therapies, as I said, the main task is the evaluation of marketing authorizations of gene and cell-based therapies. I'm not going into further detail here, but the composition, that's something I think I should mention, because as you see here, we have members from all 27 member states. We have lost the UK colleagues, unfortunately, plus Norway and members from Norway and Iceland. So that's 29, plus their alternates. And then we have in my committee 2 patient and 2 physician representatives and their alternates. So if we have full house, we're 66 people meeting once a month for 3 days at the European Medicines Agency. Now it's all virtual. On the next slide, yeah, you see the products that ATMPs that we have marketed in green, tissue engineered products, mainly cultured chondrocytes. In blue, you see somatic cell therapies for treatment of different diseases, metastatic prostate cancer, stem cell transplantation, Crohn's disease. And on the next slide, you see the 9 gene therapies that we have authorized since 2009. in very different indications, from injectable melanoma, Imlygic is an oncolytic virus, to Strimbelis, severe combined immunodeficiency of children with a rare immune deficiency. These are CD34 modified, genetically modified cells. You see Yescarta, Kymriah, and below Tecartus. These are the three CAR T cells. Tecartus, we have received just yesterday a notification from the European Commission that it will be approved. It was not approved yesterday, but it will be approved this Monday. So, and I will go in more depth based on two examples into these gene therapies, because I think these are the dominant products out of the three, and also I think of most interest to the audience here. On the next slide, let me go in some details in the two main principles of gene therapy or gene transfer. This is on the right side, the in vivo gene transfer, where a viral vector is directly injected into the patient. And on the left side, the ex vivo gene therapy, which implies that the patient's own cells are taken, for example, CD34 cells or T cells. They are genetically modified and then reinfused back to the patient. So that's the ex vivo gene therapy. And on the next slide, you see the first example. Again, left side, the principle of ex vivo gene therapy, in this case with engineered T cells. And on the right side, in some more detail, how this looks when a T cell has received a chimeric antigen receptor, has turned to a CAR T cell that has the capability to lyse a tumor cell that carries the right antigen. And to explain that a little bit more in detail, we go to the next slide, where next slide. Where I show you in more detail this complex and variable structural features of chimeric antigen receptor modified T cells. In the upper part, you see the structural features consisting in an intracellular domain with the co-stimulatory signals, the transmembrane domain, and the ligand binding domain, which is in the cases of CAR T cells that I have referred to and that we have authorized, a CD19 binding domain, the extracellular binding domain CD19. In the lower part, you see in a review from 2 years ago, but still very, very relevant, the main antigens that are targeted by CAR T cells. And these are mainly antigens expressed on hematological malignancies. You see here the CD19, that CD19 is the dominant type of antigen targeted for CAR T cells. And on the lower part on the left side, you see BCMA. BCMA is an antigen that is quite specific for multiple myeloma, another form of a B-cell lymphoma. So the CD19 targeting CAR T cells and the BCMA targeting CAR T cells have been and are developed for the treatment of CD19 and BCMA carrying diseases, and these are mostly non-Hodgkin's lymphoma and specific types of non-Hodgkin's lymphoma. On the next slide, you see that the status from apheresis of the patient to obtain the cells that are then genetically modified to infusion, to the patient treatment, is a very complex process, a very complex procedure, requires a lot of logistics, and implies a couple of steps which I would like to explain to you. So let's say a patient has been with non-Hodgkin lymphoma, B-cell non-Hodgkin lymphoma has been identified for treatment with CAR T cells. This patient undergoes apheresis. His T cells or the apheresis product is then shipped from the hospital where the patient has been apheresed to a European pre-selection site or directly to a US manufacturing site or to a European manufacturing site. When we authorized the first CAR T cell in 2018, most products were still manufactured in the United States. That's the country where the development comes from. In the meantime, manufacturing sites have been built in the European Union, for example, in the Netherlands, and outside the European Union in Switzerland. So we have now in Europe also some capacity to manufacture CAR T cell, but still the most CAR T cell products for European patients are manufactured in the United States. So the product then is apheresed, pre-selected, frozen, shipped to the US or European manufacturing site, thawed. Then the genetic modification is performed, and then the product is, in most cases, again frozen, shipped to the back to the treatment site, European hospital, and infused to the patient. Before the patient is infused with the CAR T cell, he undergoes a lymphodepleting or conditioning chemotherapy. So for the patient, 3 steps: apheresis, lymphodepleting or conditioning chemotherapy, and infusion. And after that, the patient has to be usually to stay in the hospital, and he has to be observed in the hospital because after infusion of these CAR T cells, we know that there are quite a number of side effects that can occur and that require a close monitoring of the patient in the hospital. So that's shown on the next slide. Yeah. So, and we've learned that these CAR T cell gene therapies have very specific toxicities and require a very specific risk management. Some of these toxicities are related to the mechanism of action. For example, the cytokine release syndrome. The cytokine release syndrome is a result of the CAR T cell that expands in the patient, that binds to the malignant cell, malignant B cell, lymphoma cell, and lyses the tumor cell, causing a cytokine release. So we know that 80 to 90% of patients will experience cytokine release syndrome, mostly mild, treated with corticosteroids and interleukin-6 inhibitors. Another specific toxicity is a neurologic toxicity, and another specific toxicity that is also related to the mechanism of action is hypogammaglobulinemia, because the patients usually experience a B cell depletion. You remember, we're talking about, in this case, CD19 targeting CAR T cells that then also have an effect on the normal and healthy B cells. So patients usually often experience hypogammaglobulinemia and have to be substituted. So you may ask, so with this complicated treatment, complicated logistics, supply chain, and a lot of toxicities, why do you authorize, or why are these CAR T cells developed, and why are they authorized? On the next slide, I try to, next slide, please. Yeah, I try to explain that. And on the example of CD19 targeting CAR T cells for treatment of patients with relapsed refractory diffuse large B-cell lymphoma. And the diffuse large B-cell lymphoma is an aggressive non-Hodgkin lymphoma. Untreated patients survive less than a year. They can be treated. The first line standard of care includes chemotherapy and a monoclonal antibody regimen, or high-dose therapy, and autologous stem cell transplantation. But patients that relapse and are refractory have poor outcomes and have low response rate to subsequent treatments. Response rate means the tumor responds and shrinks only in 23% of these patients, and they have a short median overall survival. And if patients have undergone 2 or 3 treatment regimens, there are no remaining standard of care options. There is therefore a high unmet medical need for new treatment strategies. And it was then found that these CD19 targeting CAR T cells offer a new treatment strategies for patients with diffuse large B-cell lymphoma, and also for patients with acute lymphocytic leukemia, for example. That's a leukemia cell type that also expresses CD19. On the next slide, I will briefly lead you through our benefit-risk considerations for CAR T cells in DLBCL. As I said, we evaluate quality, safety, and efficacy, and then we analyze in a very formalized way what are the favorable effects, what are the non-favorable effects, what are the uncertainties and the limitations. And in this case, the favorable effects are high overall response rate, high overall response rate, high complete response rate. That means the patient's tumor is gone. And durable remissions. That means the patient tumor is gone and does not come back for a certain period of time. Quality, favorable quality means that the product can be consistently produced, is well controlled, and has undergone a validated manufacturing process. Uncertainties and limitations in the clinical space are that we often, at marketing authorization and with the data submitted, cannot be 100% sure how long the patient will remain in remission. So that's the duration of efficacy that is not completely known. And as well as the survival of patients without a disease relapse. Uncertainties are also related to toxicities and the management of toxicities, and how safe use in treating hospitals can be assured. Uncertainties in the quality area are, for example, what I've shown you before, transfer to manufacturing site, cold chain topics, where the company and the future marketing authorization holder has to come up with data that and with strategies to reassure us that he can transport the product frozen from a European hospital to the United States and back, and that when the product arrives at the treatment site, where in the hospital where the patient is treated, the cells are still viable and have a certain certain release criteria, how we call them, are fulfilled. On the next slide. So it is very important that with the marketing authorization, we include safeguards and obligations for the post-authorization phase. What is included in the marketing authorization is the prescribing information package, leaflet, educational material for healthcare professionals, a risk management plan and a safety monitoring, manufacturing control and conditions for storage, and legally binding post-approval obligations to follow up the patients for safety and efficacy. So we have several options with that marketing authorization for example, we can give a full marketing authorization if we consider the data package comprehensive. This full marketing authorization is valid for 5 years and then reviewed. A conditional marketing authorization, and you've heard that probably recently in the context of the COVID-19 vaccine again, is given when the data when the benefit risk is positive, but the data are not quite comprehensive, and then this conditional marketing authorization is valid for 1 year and reviewed yearly until comprehensive data are provided, and then it turns towards a full marketing authorization. That means with a conditional marketing authorization, there is even more the obligation to the marketing authorization holder to generate and provide additional data on the product. On the next slide, we change gears towards another type of the second large group of products of gene therapies, and these are the recombinant adeno-associated viral vectors, where you can see which on the graphic on the AAV, on AAVs in principle on the right side, and on the left side a number of indications, monogenic indications, monogenic diseases for which these products are developed. And the advantage is that AAVs are not replication competent. They come in different serotypes. They with different tissue tropism. So depending on the tissue that you want to target, you can use different serotypes and you can insert different genes. So that's the other large part of gene therapies that we see. And on the next slide, I give you the example of an in vivo gene therapy for spinal muscular atrophy, with the background information that spinal muscular atrophy is the most common neurodegenerative disease in children. A disease characterized by the loss or dysfunction of survival motor neuron gene, and results in respiratory failure in those children that are heavily infected. We call this SMA type 1. It results in respiratory failure before age of 2 years of age, and children often die before they reach 2 years of age. It's caused by a homozygous deletion in the survival motor neuron 1 gene in 95% of the cases. And the survival motor neuron 2 gene, SMN2, can, but only partly, compensate for the biallelic SMN1 deletion. And so there is one treatment option, which is an antisense oligonucleotide called nusinersen, which can be administered intrathecally. But there is, without doubt, a high need for better treatment option, and these type of diseases caused by one defined gene are, of course, ideal candidates for products like AAV vectors. And you see on the next slide, what you may have encountered at the beginning of the year, where the product called Zolgensma was announced as the most expensive drug in the world. And as you see on the left side, close to marketing approval in Europe in March 2020. And also you see that large companies in the meantime, Roche, Novartis, and others, have entered the field of gene therapies and are developing these products to marketing authorization. So we are now dealing much more than 10 years ago with large pharmaceutical companies. On the next slide, yeah, you see that this is the product Zolgensma, AAV9 vector-based gene therapy, which we authorized in March this year with the therapeutic principle to introduce a functional copy of the SM1 cDNA to the motor neuron genes cells. And the indication that we granted is for treatment of patients with 5q spinal muscular atrophy with a biallelic mutation in the SMN1 gene and a clinical diagnosis of SMA type 1, or patients with this biallelic mutation and up to 3 copies of the SMN2 gene. The product is administered via single intravenous administration with concomitant corticosteroids, and we've granted, in this case, a conditional marketing authorization. On the next slide. Yeah, I've summarized what we see and what we expect in terms of in vivo gene therapies. As I've said, new treatment modalities for patients with monogenic diseases, and more than 30 of these products are in clinical development with the dominant indication of hemophilia A and B. That means these 4, 5, 6, 7 of these products are developed by different companies for patients with severe hemophilia, with the goal to achieve persistent gene-mediated factor VIII and factor IX protein expression, and thus avoid factor replacement therapy and bleeding episodes. And again, as all AAV so far, administration is by a single intravenous infusion, and if the data develop as expected, this could develop towards a practice-changing treatment for patients with hemophilia. Next slide. That's now what we have in the pipeline. We have currently 7 marketing authorizations with ATMPs ongoing, and we expect to see 10 submissions per year. That's a steep increase in what we have seen in the first 10 years of CUT's existence. Most of these ATMPs that are submitted will be gene therapies in the form of genetically modified CD34 cells, genetically modified T cells, the CAR T cell example, with a cluster around CD19 and BCMA targets in hematological malignancies. A couple of these are bispecific CARs, and the development is less speedy in solid tumors compared to hematological malignancies. The other large group are the recombinant AAVs for treatment of monogenic diseases in children. And we expect next year the first ex vivo genome editing product to come for marketing authorization. So with this, I conclude that marketing authorization is part of a complex medicine development process involving different players in the European Union. The national competent authorities in Germany, BfArM and PI, are responsible for pre-authorization activities, including clinical trial approval, and are also represented in the scientific expert committees at the European Medicines Agency level, and are there responsible for marketing authorization evaluation of what we call centrally authorized medicines. Central authorization results in a single marketing authorization by the European Commission, valid at the same time in all European Union member states. And ATMPs are the most innovative of these centrally authorized products. They are complex and innovative medicines assessed by the Committee for Advanced Therapies. And the steep increase in these ATMPs and mainly gene therapies results in new medicines for treatment of patients with unmet medical need. With this, the last slide, I would like to thank you for your attention.
Martina, thank you very much for this highly informative presentation. So I'm wondering, it is highly complex to get marketing authorization in Europe, at least. So, and if you are a large pharmaceutical company, you want to cover the whole market in the world, what would you recommend? To start at first in the United States and then in the EU, or the other way around, or at all places simultaneously?
Yeah, that's a good question. Of course, I would wish that Europe would be the best place to start these type of developments, start developing gene therapies. I would wish, but that's wishful thinking, because the products are CAR T cells, AAV vectors, are developed in the United States. They start in these large comprehensive cancer centers like Sloan Kettering, University of Philadelphia, MD Anderson, and others. That is also why the patients enrolled in clinical trials are the highest proportion US patients, because it's in the US where the clinical trial starts, and once they've started there, they come over to Europe to get their clinical trial approved here, and they come then to marketing authorization. But very often they have already submitted their marketing authorization some months before to the FDA. So still the main, these developments start in the United States.
Okay, and what about Asia?
Yes, Asia is, China is also very active, and the products are authorized there. We have CAR T cell developments coming from China as well. That is increasing. Developments in China that we note are increasing, but without doubt, Asia is very active and is very innovative, and many products are already, gene therapies, ATMPs are already available in Asia. Yes.
Okay. So there are two questions in the chat I want to ask. One is from Udo Schimmel. He asked how often on average in the process you need to ask to the submitting company for additional data.
Mm-hmm. Yeah, Udo, that's a good question. We usually have 2 rounds. That's the average. So we have the day 120. Then we ask a round of questions, we receive the answers, and then there is very often a second round of questions. So yeah, first round, second round, and third round in some cases.
Okay, there is another question from Peter Moritzen. He asked, what are EMA requirements regarding genotoxicity assessments?
Genotoxicity assessments are done in the on the non-clinical level. That means that these respective studies have to be done in vivo, usually in relevant animal models, where you, so you need to select the appropriate animal model and do the genotoxicity studies in that animal model.
Okay, two further questions popped up. One is from Karl Frese. He asks, if RCT is not possible, how will a registry-based evaluation work? And a further one, how is providing protocol assistance for MA of orphan drugs for rare diseases?
Yeah. So your first question is whether randomized is related to a randomized control trial. Is that correct?
I think so, because
okay.
I couldn't translate the abbreviation.
Yeah. So we have, as I've shown you, we have this pre-authorization phase where clinical data are generated in clinical trials, and these can be randomized controlled trials. In our cases, with these rare diseases, these are usually single-arm trials. Then when the product is then authorized in the post-authorization phase, the additional data are collected, and usually not within a formal clinical trial, but on the basis of what we call a registry-based study. So that is a systematic collection of data from patients that are treated under real-world conditions. They can be treated, they can, yeah, so that's a registry-based study. And we, in the case of ATMPs, there is always the need for a product that is licensed, and when the patient, let's say a child with spinal muscular atrophy, is treated, that his data are collected under real-world conditions. That can be done within a registry, but it can also be done outside a registry in the responsibility of the marketing authorization holder.
Okay.
I hope that this answers your question. So real-world data is
not my question, but it is
So real-world data is we talk a lot about real-world data, but they are usually it's usually the area of the post-authorization data generation, while pre-authorization we look at data generated from clinical trials.
So there is another question.
Sorry, and protocol assistance. That is done for often diseases, yeah, or drugs that have that have the orphan drug status, and you can find the information on the EMA homepage how you can submit and how you can manage to receive protocol assistance.
Okay.
Now that should be easily found on the EMA homepage.
Okay, Carl, you know what to do. So now a question from Sebastian Chirac, here from Cologne. What would you recommend to increase and foster the number of phase 1 gene therapy trials in Europe?
I would recommend that you do that you bring your entity, your drug, your gene therapy, whatever that you consider could be developed towards a medicine, that you do all the non-clinical pharmacodynamic tox kinetic experiments that are necessary, that you then translate the results to the clinic, and that you then do your first in man study in the clinic, and that you do this with the regulatory support and scientific support of us colleagues at the Paul-Ehrlich Institute, because that is our daily business, that we advise colleagues in in academia, in biotech, how to translate their research results to the clinic. And that's, by the way, one of my main interests, to help academia translate their discoveries to the clinic, to phase 1 studies.
So a final question from Yannick Delpou. Are the quality guidelines for manufacturing set nationally or by the EMA?
Yeah, the guidelines are on the EMA level. We have just finalized a quality non-clinical and clinical guideline for genetically modified cells, which will be published next week on the EMA homepage. If you would like to, if you need a specific guideline in the area of gene therapy, cell-based therapy, you can write me an email and I will get back to you. But these guidelines are developed in the scientific committees on the level of the EMA with the help of us national experts.
Okay, I think in the interest of time and because the concert will start very soon, we have to stop the discussion here. Maybe you will be available on other opportunities and occasions that discussion can be continued. It was very interesting for me.